Περνάμε τώρα στην κυρία από το Λονδίνο, στην κυρία Αγγελική Ασημάκη, η οποία μετά, ξεκινώντας από το Λονδίνο, βρέθηκε στη Βοστόνη και από τη Βοστόνη ξανά στο Λονδίνο. Είναι μια στενή συνεργάτηδα της ομάδας του Νίκου Πρωτονοτάριου. Είναι από τους ανθρώπους που συνέβαλαν καθοριστικά στη διερεύνηση της νόσου Νάξου. Την έχουμε κοντά μας.
Good evening, and thank you very much for the invitation and the opportunity to be here. I'm Asημάκη A, and I will talk to you about the molecular mechanisms of pathogenesis of arrhythmogenic cardiomyopathy. So despite decades of research, the diagnosis and management of ACM remain challenging. This is in part due to the vast phenotypic variation, age-related progression, and incomplete penetrance characterizing the disease. It is also, however, due to our limited knowledge of the pathways underlying disease pathogenesis. There is now a pressing need to understand these mechanisms so we can develop much-needed mechanism-based therapies. So what do we know about ACM at the molecular level? We know that it is primarily a disease of the desmosome. We know that it is characterized by redistribution of key proteins from junctional to intracellular pools. So the desmosomal protein plakoglobin, the major gap junction protein connexin 43, the major subunit of the cardiac sodium channel NAV1.5 and the ion channel transporter SAP97 are redistributed from the intercalated discs, the junctions, to the cell interior in the myocardium of patients with ACM, regardless of the underlying mutation. We know that there is increased apoptosis in cardiac myocytes depicted by the arrows and inflammation, both in the form of inflammatory infiltrates and in the form of cardiac myocytes themselves secreting potent cytokines. Previously, we created a zebrafish model of the disease with cardiac-specific expression of plakoglobin bearing the 2157 deletion 2 mutation, which in patients causes Knox's disease. Now, these fish exhibit bradycardia, reduced cardiac output and reduced stroke volume by 48 hours post-fertilization. Soon, they develop cardiomegaly, peripheral edema, and they show increased mortality by the time maturity is reached. We modified this line for high throughput screening, and we screened a library of 4,800 bioactive compounds for disease modifiers. One of these compounds, SB216763, could alleviate bradycardia and contractility defects and significantly increase survival in the mutant fish. And interestingly, if you take fish at 1 day post-fertilization, expose them to the drug for 6 days only in their water, and then put them back in regular water, drug-free, for an additional 7 weeks, they do not develop the disease. So this drug can prevent and reverse ARDC in fish. It also cures the disease in a cell culture model of it. Our cell culture model of choice is neonatal rat ventricular myocytes, which we transfect with adenoviral constructs expressing mutant desmosomal proteins known to cause disease in patients. Once you add the mutant vector within 24 hours, you see plakoglobin leaving the junctions and translocating to the nuclei and the cell interior. Connexin 43 leaves the junctions. You start seeing cardiac myocyte apoptosis, and the cultures start expressing and secreting potent cytokines. But if you add SB21 for 24 hours only, you eliminate each and every one of these features of the disease. What about an in vivo model? Dan Judge and Stephen Chelko at Johns Hopkins in Baltimore have created a knock-in mouse that is homozygous for a mutation in desmoglein 2. Exons 4 and 5 of the desmoglein 2 gene were removed, causing a frame shift and premature termination of translation. The resulting product is unstable and targeted for nonsense-mediated mRNA decay, which is why you see this massive drop in the protein levels. Now, these animals, they show all the features of the human disease: dilatation of the right ventricle, thinning of the right ventricular free wall, aneurysms, a massive drop in ejection fraction, 45%, as opposed to 85% in their control littermates, fibrotic replacement of the myocardium, plakoglobin and connexin 43 are gone from the junctions and have translocated to the cell interior. In terms of ECG features, there is an increasing QRS duration, reflecting more ventricular ectropy. premature ventricular contractions, of which you can see two different types here, and runs of VT. But if you start at 3 weeks of age and you inject these mice with SP2 every day over a period of 13 weeks, then you see a dramatic restoration of ejection fraction, up to 74%, as opposed to 45% in the untreated mutant mice. There is no dilatation, no fibrosis, no aneurysms or inflammation in the heart. The QRS duration is restored back to normal, and the proteins of interest, plakoglobin and connexin 43, go back to the junctions. So every feature we have encountered in every experimental model, in vitro, in vivo, ex vivo, we can eliminate with this drug, which can never make it to clinical trials because it causes cancer. It did, however, give us great insights into the mechanisms of disease pathogenesis. See, this drug is a specific inhibitor of glycogen synthase kinase 3 beta. Now, GSK3 beta under normal conditions resides in the cytoplasm together with adenomatous polyposis coli, axin, and casein kinase 2, where they form a complex known as the degradation complex. The role of this complex is to phosphorylate target proteins, earmarking them for ubiquitination and proteasomal degradation. But in the presence of ACM-causing mutations, GSK3 beta leaves the cytoplasm and now is concentrated at the junctions. And this happens in mouse models of the disease, in cell culture models of the disease, and more importantly, in patient myocardium. And it is specific to ACM because we did not see it in any other form of heart disease we examined. The fact that a single molecule can fix it all is telling you that both the cell injury and the arrhythmia phenotypes of ACM go back to one common pathway, at the core of which you find GSK3 beta. And chronic inhibition of GSK3 beta could cause cancer. But what if we looked downstream of this? And what if we could target something that is safer? Inflammation has been recognized as a feature of ACM since the disease was first described by autopsy pathologists more than 40 years ago. Indeed, inflammatory infiltrates can be found in up to 88% of hearts from ACM patients. Then patients with ACM have high levels of potent cytokines circulating in their plasma, and cardiac myocytes themselves secrete potent cytokines. Now, up until now, it was believed that inflammation is a secondary phenomenon, a consequence of the death and destruction occurring in the hearts of ARVC patients. It does, however, appear that the disease progresses through bouts of inflammation, and during one such bout, the histological image we get is reminiscent of myocarditis. So could it be that inflammation is actually a driving force of the disease? And if so, if we block inflammation, can we stop the disease? NF-kappa beta is a master regulator of inflammation. Under normal conditions, the NF-kappa beta proteins P50 and P65 are bound in the cytoplasm, inhibited by IK beta. But upon stimulation, IK beta is targeted for proteasomal degradation. The NF-kappa beta proteins are freed and they go to the nucleus, where they drive the gene expression of the immune response. There are clear links between GSK3 beta and NF-kappa beta. If GSK3 beta is active, you have inflammation through the NF-kappa beta pathway. If GSK3 beta is inactive, you block inflammation through the NF-kappa beta pathway. So this is the pathway that we targeted next. We previously said that our cell culture model of disease has a lot of features of the human disease. So plakoglobin is gone, connexin 43 is gone, GSK3 beta goes to the junctions, there is apoptosis, and the cardiac myocytes secrete potent cytokines. So is NF-kappa beta on? And the simplest way to answer the question is to immunostain for P65. So in wild type cells, then you see this diffuse cytosolic distribution. But once you introduce an ACM-causing mutation, you see the nuclei lighting up, which is a sign that NF-kappa beta has been switched on. So we treated these cultures with BA117082, a specific inhibitor of NF-kappa beta, and within 24 hours, you reverse every pathological feature of the disease. First of all, you switch off the NF-kappa beta pathway. Then placoglobin and connexin return to the junctions. GSK3 beta returns to the cytoplasm. There is no apoptosis, and the cytokine profile becomes identical to the one of control non-mutant cells. And what about the in vivo model? First of all, is NF-kappa beta on? And in this model, we answered the question with a Western blot. We looked at the levels of P65, and the total levels are the same between wild type and mutant animals. But the levels of active P65, the one that has actually gone to the nucleus, are much higher in the mutant mice, indicating that this inflammation pathway is on. So we started at 8 weeks of age, injected the animals with VA level through continual infusion with a pump over a period of 8 weeks, and we restored everything. Ejection fraction goes back to normal, the heart is not dilated or fibrotic, there is no apoptosis, the ECG features were restored, placoglobin went back to the junctions, so did connexin 43, and GSK3 beta returned to the cytoplasm, and the cardiac myocytes stopped releasing potent cytokines. Now, we mentioned briefly that inflammation is dual in ACM. On one side, you have the professional inflammatory infiltrates. On the other side, you have the innate immune response of the cardiac myocytes themselves. Now, in a model like the mouse, you do not know which one of the two components is the one driving the disease, but we can make cardiac myocytes from induced pluripotent stem cells obtained from a patient. And these cultures are more than 95% pure myocytes. There are no macrophages or other professional immune cells in those cultures. And you see that NF-kappa beta is beautifully on by all the nuclei lighting up, and these cells secrete a ton of cytokines in their cell culture milieu. But you add BA11 for simply 24 hours, you switch the pathway off, and you restore the entire cytokine profile back to normal. So to conclude, we showed that NF-kappa beta is on in 3 different models of ACM, and we showed that blocking it can mitigate the disease. And as a matter of fact, it appears that the immune response from the cardiac myocytes themselves is more important in driving the disease than the professional infiltrating cells. Much more work needs to be done, but this study introduces for the first time the possibility of anti-inflammatory mechanism-based therapy in the management of arrhythmogenic cardiomyopathy. Thank you very much for your attention.
Thank you, Angeliki. Thank you very much. It was impressive, informative, and exceptional. You don't hear? Can you hear me? Can you hear me? Έχουμε ένα μόνιμο. Can you hear me now?
Σας άκουσα. Εσείς?
Yes, I said that we had a very informative, impressive, and exceptional presentation. Can you hear me now?
Thank you very much.
All right. So how far are we from the drug?
Since we have identified the pathway, it is now a matter, in my eyes, of trying repurposed drugs. So drugs that are perhaps out there already being used either as FDA-approved drugs or in clinical trials for inflammatory conditions, but that we know they target the same pathway. And if we go down this road, we are much closer to actually bringing a drug to ARVC patients than if we start from a chemical from scratch. So we could be talking for in under 10 years and with a much lower cost. So that's what we are working on at the moment. Drugs that are already out there, which we need to try them on more in vivo models to make sure that the effects are not mutation specific. And then hopefully one day we can bring them to the clinic.